Evan Henry, our next speaker. Evan and I have known each other for a very long time. Evan has been my patient, but also a strong spokesperson for Parkinson's disease and neurological illness in general. He is a husband, a father, continues his full-time employment and still finds time to do things like write a novel and jog and participate as a patient advocate for the stem cell programs, not just here at UCI, but also regionally. And we have, I think, through Evan, a national impact on advocacy for neurological illness. So Evan.
Well, thank you very much, Neil. I'm not sure that I live up to all that bright and shiny introduction, but I'll do my best for 5 minutes. I'm a member of the Patient Advocacy Committee here for the UCI Stem Cell Research Center. I've been on the committee for quite a few years with some of my compatriots also in the audience. Nan Luke and Francis Saldana and Beth, sort of blanking on her last name, but she's one of the seeing eye dog here today. Koenig, that's right. Talk about that mind slipping, that little bit of memory recall stuff problem. But what I really am is a stem cell groupie. And what I mean by a groupie is that when they were moving into this building, I offered to carry the equipment and set up a lab for them. I also have been known to make up wild stories about the after research parties here, especially when there's press in the room and I get to be interviewed. I recently thought, if Neil Hermanowitz takes some skin cells from me, I'm going to get a stem cell tattoo to go over the top of it to hide the scar. But I figured, sort of memorialize it in something pretty as opposed to something not so pretty. I think the only thing that I haven't done is that there's no reason to play a cowbell here, like I would in a band, or if I accompany a band as band grouping. I can tell you though, I like the science. I go to a lot of the lectures here that they have. They bring in a lot of visiting lecturers, whether it's from other institutions, the people who are doing exciting things here, or even the NIH. I learned a lot about bone cells the other day. Some of the things that I've actually starting to understand include what we've seen today is that there's actually a difference between oligodendrites and astrocytes and neurons and neuronal cells, because neuronal cells are the ones that turn into all those other three things. So you start to understand the actual basic science. So when they start talking about things like BDNF and GDNF, I know the one is the brain-derived neurotrophic factor and the other one is the glial-derived neurotrophic factor. Now, I haven't quite figured out what the difference between brain and glial is, but I think I got a clue today. So it's actually stimulating to come here and listen to those kind of talks. I could talk about being a patient, having Parkinson's disease, diagnosed for 8 years, how it took 2.5 years to get diagnosed. The funny thing was I lived 2 miles from here, and it took me 2.5 years to get 2 miles to get to Neil's office to be diagnosed. But that's fairly typical also in the case of a lot of these diseases. They're very, particularly in young onset, they're very hard to figure out if you don't go to the right people. But you don't really need any more stories. I'm sure everybody has their own story, and frankly, I think mine is fairly unremarkable. But what I think is remarkable is the things that I can do by being part of a place like this. We're doing really remarkable things on the development of just understanding the possibility for new cures or new treatments, and they're actually even gone to human trials in things like the spinal cord recovery and injury repair. That's just really exciting. It was really fun. I was at my mom's house this last weekend in Connecticut, and I read about a story about the study that is ongoing in Georgia, based on the work of Hans Kierstedt from UCI on spinal cord recovery. It was really exciting to see that it got that kind of national press, and I read about my own backyard and my mom's backyard in Connecticut. But the thing that's remarkable, I think, is that we can participate in things like clinical trials as patients and patient advocates. We can be involved in outreach to other people who are in need and need to discover things like this. We can be involved in fundraising. And one of the things that I just like to do is I like to hang out with the researchers and just tell them thanks, because I'm really excited about what they're doing. They're going to do it for me. One of those remarkable people is somebody I get to introduce now. I hadn't met her before, but she's doing some really exciting things, particularly with Parkinson's. And that's Dr. Birgitt Schuele. She's the assistant professor of clinical molecular geneticist at the Parkinson's Institute in Sunnyvale up in Northern California. She has her MD degree from Medical University Lübeck and her doctoral degree from George August University, also in Germany. We're fortunate that she came to the United States 8 years ago for a 1.5-year postdoctoral fellowship at Stanford and decided to stay on. And so without further ado, I'd really like to introduce you to one of my heroes in this kind of research based on what I've heard she's doing, and I'm really excited to hear from her firsthand. So Birgitt.
So it's really a pleasure to be here at this special day for patient advocacy funded by CIRM. I have the opportunity to talk about Parkinson's disease and stem cell research. I have divided my talk into 3 parts. The first is, and some of you might not know what is Parkinson's disease. Some of you already talked at the booth and know very well what Parkinson's disease is and what they can do today to fight it. So I talk about what it was in the past, and how do we see Parkinson's today. The next part is, how do we treat Parkinson's disease? What are treatment options today, and what are treatment options for the future? And then the last part is, what are new avenues for stem cell research? And I'm going to talk about Parkinson's disease in a petri dish. The textbook definition of Parkinson's disease is that it's a slowly progressive neurodegenerative disorder that was first described by James Parkinson in 1817. It affects about 1% of the population over the age of 60. Key symptoms are slowed movement, stiffness of the body, shaking, and gait and balance problems. The pathological correlate there is that we see a loss of dopaminergic neurons in the brainstem, and that's depicted here. This is a brainstem. This is the substantia nigra, or black substance. And in Parkinson's disease, you see this very pale structure. So we're losing neurons in the brainstem. What we also see is something that's very characteristic for Parkinson's disease. It's called Lewy bodies. These are protein inclusions that we find in the brain of Parkinson's patients. But this is not all. The substantia nigra and the motor symptoms of Parkinson's disease is just the tip of the iceberg. We've learned through research studies, clinical research, pathological studies over the last 20 years, that there's much more to it than the substantia nigra. There are other brain regions that are affected. And now we also know that the peripheral autonomic nervous system is affected. And we see these protein inclusions that are called Lewy bodies all over the body. So it's a systemic disease, and it's not just confined to the substantia nigra. Initially, we thought it would be a good start for stem cell therapy because you just have to replace those neurons in the substantia nigra. But now we learn it might be much more complicated to attack this disease. So these are the non-motor symptoms that are adding to the motor symptoms. Since we have these changes in the whole body, we also see cognitive changes like depression, anxiety. We have sleep dysfunction in Parkinson's patients. We have orthostatic hypotension. A lot of patients complain about constipation or heart rate variability. I just talked to Evan, and he was talking about his way when he's jogging, that all of a sudden the heart rate drops. A lot of patients also have a loss of sense of smell, which can be something that even precedes the symptoms of Parkinson's disease. So to summarize that, for over almost 200 years, we considered Parkinson's disease really as a localized loss of brain cells in the brainstem. But over the last 20 years or so, maybe a little longer, new research really showed that it's a systemic disease with really widespread pathology over the brain, heart, intestine, and this shows characteristic non-motor symptoms. So the view of Parkinson's disease has changed over time, over the last 20 years dramatically. What are our treatment strategies for Parkinson's today? So this is just a timeline, and you see I have in a red box L-DOPA. Levodopa, the dopamine replacement that we need for treatment of Parkinson's disease. It's a symptomatic treatment, and this is still the #1 treatment option. There are a few other drugs that are coming on the market, but I think we need really innovative drug discovery to find better drugs that halt the disease or even reverse the symptoms. I wanted to mention deep brain stimulation. This came up in the 1990s. This will help a subset of patients, not everyone. The other thing that I discussed at the booth this morning, it's not just the drugs that you need. You can do something today without any prescription. You can exercise, you can eat healthy, you can do yoga. There's so many things that you can do to improve your life and your symptoms today. I wanted to mention that. But again, we need better drugs. Since this is a stem cell meeting and CIRM is sponsoring this, I think Parkinson's disease was one of the first diseases where there were really courageous clinicians that wanted to cure Parkinson's early on. And it started in the 1980s with adrenal medullary transplants, first trials of fetal neurons pioneered in Sweden and Mexico. And then over a period of about 10 years, there were open label trials using fetal transplants in Parkinson's patients to cure the disease. However, I think that was a little premature. And even though we funded 2 double-blind placebo-controlled studies by NIH, these trials failed, unfortunately. The other thing that I wanted to mention is in 2008, there were a few papers that were coming out that were looking at autopsies from patients that received these fetal tissues. And what we've seen in the brain that these neurons, these fetal neurons, show Parkinson's pathology. So this just should show us that we should be really cautious jumping into those therapies. Even we know it's really urgent and we want something now. Some of these patients that received these transplants really suffered and they had severe side effects that needed additional surgery. So I'm not saying we shouldn't do it. We should do it safely and carefully and understand the disease before jumping into it. This is the neural graph that was affected with Parkinson's disease. So what we see is, even though we transplanted healthy cells into the brain of Parkinson's patient over time, over about a decade, this graft got Parkinson's disease, and this is a Lewy body within the graft. I think now we have better techniques. We have now human embryonic stem cells that are in the pipeline, and I really hope that they will offer a new source of tissue for stem cell replacement. I know we have now better controlled culture conditions. We can also produce a higher yield of the target cell population, which is the dopamine-producing neuron. And we also can use human embryonic stem cells as vehicles to deliver additional proteins. I'm not going into detail here. My talk should then focus on iPS cells and how do we make Parkinson's disease in addition. Okay, to summarize my second part, current drug therapy is limited. We still have L-DOPA after 40 years, and it's still the main treatment option for Parkinson's disease. So I really think there's an urgent need to find new therapies and new drugs. The fetal neuron transplantation was a very courageous step in the 90s, but we had two major drawbacks. There were severe side effects in a subset of patients. You can also see it the other way around. There was some that had significant benefit, but we can tell which patients really have that benefit. And also the graft tissue can get affected with Parkinson's disease. So really hope that embryonic stem cells offer an alternative to that. So now to the main part of my presentation, Parkinson's disease in a petri dish and new avenues for stem cell research in PD. We are funded by CIRM. We also received an early translational grant to carry out this research. And we make patient-derived, human-derived induced pluripotent stem cells. We are starting with a patient. We are starting with a skin biopsy. Bank those cells, and then we do something that's called epigenetic reprogramming, which is a new technology, and it's amazing. What we can basically do, we can rejuvenate cells and get them back into a pluripotent state. I had to convince my mom, she's a biology teacher, that this is possible, because it has been a dogma that once a cell is differentiated into a tissue type, you can't go back. You can't turn the clock backwards. Now we can. And the people who pioneered that might receive the Nobel Prize for that. So this is a very exciting technology that we now use. And it's pretty new, but the good thing is it works. A lot of laboratories are using this technology now, and you can model a lot of diseases in the dish now. So once you have the pluripotent stem cells and you've characterized them well, we are interested to make neurons, and we are interested to specifically make the dopamine-producing neurons. And then there are two avenues: the transfer back into the brain, so the cell replacement, and that would be just a phenomenal way to use those cells. However, as I've shown you, there are some caution that we have to take before this is a valid treatment option. The other one is in vitro research, and I'm very excited about that avenue. What we can do now in a culture dish, we can model Parkinson's disease from a human individual that had Parkinson's. Disease research, disease mechanisms, and we can also use those cells for drug screening, which is very new and very exciting. So replicating Parkinson's in disease in the laboratory. The problem to date was that there was no really cellular and animal model to replicate every pathological feature of Parkinson's disease. You could replicate some, but a lot of the cellular models that we've used were from cancer cell lines, and we tried to understand disease mechanisms of Parkinson's disease, which doesn't really work out. So we rethink that patient-derived stem cells that we can make in the culture dish right now hold a promise to become an authentic model of Parkinson's. So how do we rejuvenate our skin cells? We start with the skin cells in a culture dish, and they look like this. These are these long, elongated little structures. We are adding specific factors into those cells, and you need between 1 and 4 factors, and there are different techniques how to make them. But you can reliably get from skin cells into induced pluripotent stem cells, and you see just from looking at these two images, they change morphology quite dramatically. So you see here these aggregates of cells. These are maybe a couple thousand cells, and from one of these little cells, a stem cell colony derived. It's just an amazing process. We also have to make sure that these induced pluripotent stem cells are good. So we have a quality score chart. We have to do a lot of assays to make sure that these cells are really very close to human embryonic stem cells. We look at markers, we look at at the ability to differentiate into different tissue types of the human body, and we have to do some karyotyping, epigenetic changes. That's way too much for this audience, but it's very important for reviewers that we prove that these are really stem cells. So what we want to do is we want to study neurons, dopamine-producing neurons. And the question is here, how do we make nerve cells? There are many protocols out there, and what we're using is we're using an embryonic body technique. We're starting with a stem cell colony in a dish. We modifying the nutrients in the feeding media and culture those cells as floating little aggregates for about 10 days in culture. After 10 days, we are plating them down onto coated culture dishes. And then after a few days, after 4 to 5 days, these embryonic bodies form structures that we call neuronal rosettes, or rose-like structures. I look at them, to me they look more like a cauliflower. So, but I guess rose-like sounds better. And then manually you have to go in with a little syringe to cut out these structures that are these rosettes. And then we expand them as single cells and plate them down in another dish. At this point, we call them neurostem cells, and they again have to express a certain set of markers. In the final differentiation step, what we do is, and that takes about a month, we are adding other recombinant factors and signaling factors. And here, again, you have BDNF, GDNF. Those are factors that are important to differentiate the neural stem cells into dopamine neurons. On the next slide, you see how the dopamine neurons look in a petri dish. And this is a very nice network of cells that have been differentiated from induced pluripotent stem cells into neurons. In green, you see a neuronal marker, and in yellow, you see the cells that are producing the dopamine. So now we have the cells in the dish with which we can study Parkinson's disease. When we started this grant, or when we discussed ideas in our collaborative group, reviewers said, hey, you might not even see a difference, because what you have here are basically neurons that just became neurons. They are baby neurons. And you want to study a late onset neurodegenerative disease. That will never work. So to convince those people, we can now show that there are differences between the Parkinson's cell lines and control lines. In this case, what you see here is a patient on the left side in this panel who is carrying a specific Parkinson's mutation. It is a multiplication of a gene that we think is associated with Parkinson's disease. And what we see is, in this carrier, he's carrying much more of this protein that is depicted here in green compared to the sibling control line. We also quantified that by a different method, and what you can see here, the relative protein amount is about twice as much as in these two control lines. This is a paper that's in revision, and we definitely see a difference. This is a paper that just got published a month ago in Cell Stem Cell. And what we can show here is that the Parkinson's neurons, the new neurons that we get in the culture dish, are more vulnerable to oxidative stress. So you don't have to understand this complicated slide. I just want to point out to these blue bars. These are the Parkinson's lines. And the gray bars, these are the control lines. And what you see here is when we are adding a stressor to those cells, in this cell death assay, the Parkinson's line is doing much worse than the controls. So we already have an assay in the culture dish that can distinguish between Parkinson's and controls. And these are the potential applications for drug screening. We're starting with patient fibroblast, going back, rejuvenating those cells into induced pluripotent stem cells, then going back into dopamine neurons. And now we have assays, and we can actually see a phenotype. So now we're in the position to say, okay, let's test drugs. Let's test drug libraries. And that's what we are hoping to do in the next year or so, to miniaturize those assays and have them ready for a drug screening format. And then further down the road, you go into preclinical and clinical development, and hopefully at some point to the market. So the hope here is to find new drugs that are not just L-DOPA equivalent, that can halt or even reverse the progression of the disease. To summarize Parkinson's disease in a culture dish, I think the patient-derived induced pluripotent stem cells really provide an entirely new, and I hope really better model to understand Parkinson's disease mechanisms. And I also think that these tools will help to find new drugs for Parkinson's. With this, I want to thank a lot of people here. First of all, I want to thank all the patients and volunteers participating in the studies that have given skin biopsies for this study. It has been an enormous success in our clinic. Everyone would like to participate because a lot of patients see the great potential that we have with these induced pluripotent stem cells. I want to thank Dr. Langston, who is a leader at our institute and who has been the principal investigator on this grant. All these people in the lab have spent a lot of hours deriving those lines. Once you have an induced pluripotent stem cell line, these are your babies. Oh, sorry. And you have to care for them 24/7. So they don't take a vacation. You have to come in on the weekends to feed them. So you have to have a feeding schedule to keep them happy. And I also want to thank all the collaborators. And you see it's a big group of people that is interested, from academia and also from industry. And specifically at the Stem Cell Institute at Stanford, there's Renee Reopera and Dr. Theo Palmer, who are collaborators on this early translational grant. I want to thank them. And without the support of CIRM, I wouldn't be here. This program wouldn't be there. It's just a great success, I think. We got additional seed support from the Bloom Foundation and the Parkinson Alliance. And I want to thank you, all of them, for your attention. Thank you very much.
Lunch is looming. Does anybody have any questions for Dr. Schuele before? Yes, Francis.
I have a question.
Yeah.
You mentioned something about the olfactory nerve being sort of like declining in people with Parkinson's. But I noticed with my daughter who had Huntington's that the olfactory nerve was extremely sensitive.
Okay.
And not only with her, but with my other daughter as well. Is there any reason why it should be that way with one disease and the opposite with another? Or have you ever heard any other?
Maybe you have an answer.
It's a well-known phenomenon that sense of smell is impaired in Parkinson's disease very commonly, and perhaps even long before the motor symptoms become apparent. I don't think it's really been studied so much in Huntington's disease, so I don't think there is an answer to your interesting
So
question.
I noticed it with two patients already, my two daughters, so I'm wondering if it's common, but it's
Well, we need
extremely
to
sensitive.
we need to look.
Don't know.
There's evidence in Parkinson's of early pathology in the parts of the nervous system that are involved with detecting odors, but I don't know that's been looked at specifically in Huntington's disease. Somebody had a question in the back? Yes.
I wondered, you used the term oxidative stress.
Mm-hmm.
What is that?
Yeah, the question was, what is oxidative stress? Oxidative stress is something that is challenging the neurons, and they have to fight against this oxidative stress. So those could be toxins in the environment that can lead to it. And so the Parkinson's neurons, they don't have enough shielding to fight that oxidative stress. Okay.
One more thing, I wondered if you, on the slide that was close to the end, you were talking about you were hoping to move into this phase that was just before perhaps moving it to clinical trials. Do you have any timeline you're, hoping for on that?
Yeah, no, I think we're getting those questions, and I see there's an urgency for finding new drugs. If I would know the timeline to this, I would be much closer to better therapies. And I don't want to put 5 years to it or a shorter time frame. I will just probably lie to you on that. We're working hard. That's what I can say.
From where I sit, somebody who does a lot of clinical trials, this is what we just saw, and also with Matt's presentation, is this is a way to accelerate therapies, because there's this big step, as I kind of alluded to. You do mouse models, you do safety information, then you move into people. But using stem cells in the laboratory, as Dr. Schuele described, is a way, I think, to accelerate that process, so we have maybe more information before we move into people. That's what I was kind of alluding to with my hopefully not too cynical comments this morning, is that we move into clinical trials and we're using information from mice and rodents and then safety from people, but then we do these big, long, expensive trials that we might be able to skip if we had more information from the stem cell preparation.
That's true.
Taking antioxidant, does it help for that situation that you just mentioned?
We hope so. So, okay, taking antioxidants, would that help with the shielding of the neurons? At least in tissue culture and in animal experiments, yes, it helps. You mentioned the CoQ10 trial. That was less encouraging.
It was discouraging, unfortunately. It's a great idea, and again, there are things that are very attractive in rodent models and tissue culture, but not tissue culture meaning cells in the petri dish, but not like we've seen presented here. Not really the human cells derived from people with Parkinson's disease or with Huntington's disease. This is really a new way of looking at and developing therapeutics. We should wrap up for lunch.
So I'd like to thank all the speakers this morning. They did a great job. Thank you very much.